Biology, Part 92. The most important organ of temperature control in mammals is the skin. The structure of human skin is described in the next section and must be understood before the physiological mechanisms of temperature regulation in humans and other mammals can be usefully discussed. Human skin. The skin of an adult person has a surface area of about 2 millimeters square and varies in thickness of 0.5 mm in most body region to 3 mm on the soles of its feet. It is tough and flexible and provides mechanical protection for the underlying tissues. In addition, it is major sensory surface, manufacturing vitamin D, screens the body from harmful ultraviolet radiation, and prevents the entry of bacteria and other microorganisms. This defense function is described earlier in our tutorial. It plays an essential part in the temperature regulation. Two distinct regions are the epidermis and the dermis, are easily recognized by the vertical sections of human skin if we see it in the figure. Epidermis. The outer epidermis is firmly cemented to the underlying connective tissues dermis and itself consists of several cell layers. The much folded stratum basale consists of a single layer of cells which remain capable of cell division. Division results in cells which are pushed upwards and gradually pass through the remaining layer before being shed at the skin surface. Together, the stratum basale and the stratum spinosum, which contain 8 to 10 layers of the cells from the germinative layer of the skin. This is followed by a third cell layer of the skin, and that is called stratum granulosum, which consists of several rows of flattened cells containing granules and caroteolin. The nuclei of these cells start to disintegrate at the caroteolin is converted first into elidin and then stratum lucidum and finally into the waterproofing protein keratin, which fills the cell from the outermost stratum corneum. The flat dead cells of the stratum corneum form flakes and squames which are continually shed. A large proportion of household dust consists of human squames, and it is estimated that human epidermal cells take approximately 27 days to move from the stratum basale to the surface, so that depending on its thickness, the entire epiderm can be replaced in this time. The color of the skin is due to cells called melanocytes, which are located between the dividing cells of the stratum basale. The black pigment melanin produced by these cells is taken up by the cells in the remainder of the epidermis, helping to screen out ultraviolet radiation. The number of melanocytes in people, different races, is approximately the same, but they are much more active in black and colored skinned people. The amount of melanin produced is increased by the melanocyte stimulating hormone MSH, but the importance of this is not fully understood. Dermis. The inner dermis consists of a connective tissue matrix containing collagen and elastic fibers. Its upper region is called papillary region, gives rise to the finger-like projection, the derma papillae, which extends upward into the epidermis, and the papillae contains capillary loops and touch-sensitive nerve ending. Within the remainder of the dermis lie numerous structures including sense organs, nerves, blood vessels, lymph vessels, hair follicles, and glands. Some of these are described below. Number 1. Sense organs. The sensory structure present include free nerve ending sensitive touch, pain, and temperature. The Meissner's corpuscles sensitive to touch, and Pacinian capsules sensitive to pressure changes. The distribution and dentistry of sense organs varies. They are most numerous in the lips and fingertips. and spots in the regions like forearm or shoulder.